Well, as Matt said, I'd like to visit with you today about an approach that we're taking towards reducing the cost of genotyping. And so a quick overview of the presentation. I will start off by discussing a concept called mass genotyping by sequencing technology and explain to you what that is. Go through how we've validated it and think about some potential applications for it. And then I want to introduce the concept of a sparse genome scan, which is somewhat similar to what Matt alluded to, but we're going to take it a little bit further than that. And then we'll talk about some applications of a sparse genome scan. So, mass genotyping by sequencing technology is an approach to low-cost, high-volume DNA testing, and it's based on recent developments in next-generation sequencing. Next-generation sequencing is a technology that's used in a lot of areas of science and biology. It's a very high throughput, low cost way to generate DNA sequence information. And with that technology, we could generate the sequence of anywhere from around 30,000 to 240,000 very short pieces of DNA for about $1 of sequencing cost. So that's an enormous amount of data at a pretty low cost. Now the caveat to that is that to put a sample on the machine is going to cost around $1,000 or somewhere in that ballpark. So we're not going to do that for just one animal at a time. We've got to be able to do that for a lot of animals to spread that cost out over a number of animals to make this worth doing. So we've developed an approach towards doing that and worked with a company called Eureka Genomics through a cooperative research and development agreement. And they have taken this approach and made some modifications and improvements to it and commercialized it. And that's what I'm going to visit with you about for the most part today. So the way that works is that for each animal to be tested, we add what we would call a DNA barcode to the DNA fragments from that animal. And that DNA barcode is just additional bases of DNA sequence that are unique to each animal and that get read by the sequencing machine in the process of sequencing the rest of that DNA fragment. So it's a chemical way to add to a fragment from an animal a piece of synthetic DNA that tells us which animal that DNA originated from. So we take samples from many different animals and pool them together, run them on the next generation sequencing machine. Each fragment's read by the machine and indicates which animal the fragment's from, and it will read one of the two copies of a particular marker that's possessed by that animal. You know that each animal has two copies of each gene, and we can only read one of them at a time. So we need multiple reads per animal, per marker, to ensure that we've covered both of the copies of DNA in that animal's genome. In the process of doing that, we also discern what the position in the genome of each of those fragments is. So that allows us to know which marker we're looking at. And as I said before, we'll read each one of them multiple times. These are three different machines that could be used for this process. There are others that could be used for this process. The one up here is a Roche 454. This is a Illumina genome analyzer, and this is the Ion Torrent. We've worked primarily with this Illumina machine and several different varieties of the Illumina sequencing technology, and our colleagues at Illumina have tried out the Ion Torrent. Each of them has advantages and disadvantages. The data I'm going to talk to you about today was done on the Illumina platform, and that's where most of our emphasis is, but for particular applications where we'd need very fast turnaround, for example, we might look at something like the Ion Torrent instead. This slide kind of illustrates the general process, and I'm not going to go into a lot of detail on it. This is a very, very microscopic view of what happens on the surface of a essentially a microscope slide that has these different lanes on it. This one has 8 lanes on it. Each of those needs to be filled in order to run the machine, or at least we might as well fill them because we're going to use that capacity. And so one of the constraints is we need to obtain the same number of bases of DNA sequence on each one of these lanes when we do a run. And that places some constraints. We would optimize our run a little differently if we were doing it just to genotype than if we have one lane that's for genotyping and others that are being sequenced for other reasons. But we get about 30 million reads per lane on this GA2 machine from Illumina. And the way that it's read is looking at this under a microscope. And you can see we have quite a bit of magnification here. And the color of each one of these little clusters as we're reading each base indicates what that base of DNA is. So it takes a while to go through reading this on this machine, but it just generates an enormous amount of data at a very low cost, and that's the point that's salient for what we want to do. So another aspect of applying this technology to genotyping in the way that we're doing it is that we use chemistry that allows us to focus the sequence data on a specified set of informative loci. There are other approaches to using this technology to genotype that say we're just going to take whatever fragments come to us and do what we call a reduced representation. But there you don't get to focus on specific loci, and so that's one of the key aspects of this approach. And we would put at least 1,000 animals in a lane, pool them together, and then acquire the sequence data and take it all apart to turn it into useful genotypes. So at the Beef Improvement Federation, our colleagues Eureka rolled out this technology, and they said that they felt they could do this for $5 per animal for a panel of parentage markers comprised of about 95 markers. And so that's $5 in addition to the DNA extraction cost. But we feel that's a fairly competitive price for a panel of that size. They thought they could turn that around in 2 weeks. But this is what the price is in the startup phase, where we're dealing with low volumes and trying to get established. There's an awful lot of inefficiencies involved in doing this in kind of a low volume basis. And so they also said that if we really could get up and running to do this on a larger number of animals on a routine basis, so in other words, if they could fill one of those machines each week, they could probably do this for about $250 per animal plus DNA extraction. So we're getting down into the range where just getting the DNA out of the tissue is beginning to become the limiting cost factor in applying this technology. We also think that there's a lot of scope to increase the number of markers that we run in the assay. We're working with a set of just under 100 right now. They have done runs with a bit over 100 and without any problems, but in adding markers, it's not multiplicative. There's a fair bit of overhead in just doing the process, and then the cost of additional markers is quite a bit lower than what you see here. So we're working to see how many markers we can get into an assay before the cost becomes prohibitive. It's not, as it stands today, it's not going to be competitive at, say, 50,000 markers. It would get more expensive than a 50K chip. But for sets of maybe 1,000 to a few thousand markers, it looks pretty promising. So I think I just said most of what's on here. So we want to be looking at thousands of loci per animal. It's not definitely not 10 times the cost of 100 markers. We think there's a lot of strategies that could allow us to increase the number of animals per lane as well as the number of markers, but there's some diminishing returns if we put too many more animals on a lane. We just really don't save all that much cost. So the real improvement in the technology as we go forward is going to be more markers at price that's similar to somewhat just probably minimal increase in price. Okay, so we've validated that on data that we collected here with on animals that were produced here with the Illumina 50K chip that I think you're all familiar with. And we use these 95 SNP from Mike Heaton's SNP Parentage panel, all of which are on the 50K chip. So we validated the Eureka's results against 50K genotypes. We used all the genotypes at these SNPs for which the 50K chip gave us a call. That none of these genotyping technologies give you a usable genotype on every marker for every animal, so we used those that we had. And in the genotype calling algorithm, that's what takes the number of counts of reads that say allele A versus allele B at each of these markers for an animal. We have to use an algorithm to convert those counts into a called genotype, or in some cases we decide we don't have enough reads to give us a call. And we tuned that algorithm for this parentage application. So that means we emphasized accuracy over call rate. If we'd been more concerned about having a really high call rate and could tolerate a little bit more errors in our calls, we could have brought the call rate up at the expense of accuracy. But for parentage, accuracy is very important. You don't want to make a false exclusion because that has undesirable consequences, as those of you who have done paternity testing in Seedstock can allude to. So these are the results that we got. These are sorted by marker, from the markers that gave us the greatest concordance to those that gave us the lowest concordance. You can see even at the bottom of the list our concordance was pretty good. Those that had low concordance dropped off quite a bit for call rate. And basically what was happening here is we just didn't get as many data points for these markers as we did for these markers. But overall, and I'm sorry it doesn't show up very well here, this says the overall concordance rate was 99.1%. The overall call rate was 96.1%. So we got a result 96% of the time per marker per animal. That's not 96% of the animals, it's per marker per animal. And then the accuracy was quite good at 99.1%. This is looking at the same data, but here sorted by animal based on concordance. And you can see again at the top of the list our concordance values are quite good. Even though it looks like we're halfway down the list here, we're actually about 80% of the way through the list when we get down to about 99% concordance. And then it's right at the end that it falls off pretty badly. And we realized in retrospect there was something we should have done a little bit differently. We should have thrown out animals where we didn't have enough reads per animal, and we didn't do that. We should have dropped out probably these last 20 animals. And if we'd done that, our concordance would have gone to 99.4, our call rate would have gone to 97.1, although we would have lost 2% of the animals to accomplish that. But so we just had a couple percent of the animals that we dealt with where apparently the DNA sample just wasn't good enough to really give us good results here because the numbers of reads really fell off badly. But overall we were pretty satisfied with those results. And I should mention that these concordance rates include any errors that were present on the 50K chip. And we know that none of these genotyping approaches are perfect. So we don't know what part of that 0.6% was an error in our data versus an error in the 50K. But all in all, we had what we thought was a very acceptable error rate out of the approach. So I just said that. So the ways in which we can envision this being used are obviously for parentage testing, and that application is ready to go, and Eureka is doing that for several entities. Both parentage verification and paternity determination. The second was a little more challenging than the first. Certainly it could be used for at least some types of genetic defect testing for single gene traits like color, poleness, the myostatin F94L mutation, etc. We could use it for prediction of economically relevant traits from small panels of SNPs. And I think there's opportunities for discovering new recessive defects or for genotyping new recessive defects that are discovered through individual bull sequencing, and we're getting reasonably serious about that, and I think we'll be learning a lot out of that in the near future. So we also could apply it to the commercial segment of the industry if we can keep the cost low enough and get enough information out of it. And so certainly in the feedlot setting, there have been some pilot projects done in the past where large cattle feeding companies have used marker-assisted management from small panels of SNPs. And I think the challenge that they found there is how to get enough value, basically how to get enough predictive value from a relatively small number of SNPs. But I think as we get the genotyping technology considerably less expensive than it's been in the past, I think there's real scope for application in that way. And then many of you are familiar with the commercial ranch project that Dr. Pollack did when he was at Cornell. It basically showed that on large commercial ranches, if they would paternity test at least some of the calves produced on the ranch, calves produced by the younger bulls, calves produced the first year that a bull was used on the ranch, that they could gain enough value by having an idea of the breeding value of those bulls, an enhanced breeding value relative to what could be produced as a non-parent, that could actually pay for the cost of paternity testing. And I think if we think about that in the context of a bigger system and think about the benefits that could have to the seedstock industry in terms of feeding data back into the seedstock industry, improved genetic selection on the commercial ranch, improved management on the commercial ranch, that there's probably enough benefits there to cover costs before too long. Maybe not exactly today, but I don't think we're too far away from that being a plausible scenario for us to consider. So I want to use the few minutes that I have left to talk to you about sparse genome scan genotyping. This is the idea that if we took about somewhere in the neighborhood of 1,000 to 5,000 markers spread evenly across the genome, that we could track inheritance throughout the genome of a pedigreed population. The objective of doing that is to provide an inexpensive replacement to most of the other DNA testing that might be done on seedstock cattle. But to make it work, and to work effectively, it needs to be applied to a whole herd, or better yet, to a whole breed, or maybe to the majority of the elite seedstock programs within a breed. But it's not something you do on one animal and you take that animal by itself and say, now what can I do with these genotypes? That's the mentality that gets us to the number Matt quoted of about 10,000 markers in order to impute 50K and high density genotypes down through to the individual animal. Here what we're talking about is the idea that because we're doing dense genotyping, dense throughout the pedigree rather than dense throughout the genome, we have the opportunity to pick up an awful lot of information But it's not a one animal at a time operation. It's a matter of looking at an entire program or entire population. We don't have the technology worked out yet to be able to tell you, here's what it would cost to do 1,000 markers, or here's what it would cost to do 5,000 markers. But I'm pretty confident that in the reasonably near future, we're going to be able to get to a point where we can do this at the level that it would be cost effective for programs to genotype the whole herd instead of just cherry picking a few animals to genotype. And that really changes the scenario in terms of how we use genomics in cattle breeding. And I think this is a technology that has a lot of opportunity in the seedstock industry, less so in the commercial industry, because there we're not going to be genotyping multiple generations and tying those multiple generations together. Right? And when I talk about genotyping densely through the pedigree, I'm talking about multiple generation genotyping. And so that's where I think that those of you who've been putting away tissue for several years are going to have a leg up, is that we can go back and apply this to those tissue samples that are already stored, and I think there's going to be a lot of power to come from that. This is kind of a picture of how I'd envision sparse genome scans working. These animals have the sparse genome scan across one chromosome of the genome, and these, which are primarily bulls in this pedigree, have the 50K chip run on them. So the sparse genome scan doesn't work just by itself. It works in concert with the 50K chip, and eventually with individual animal sequencing on influential animals up at the top of the pedigree. And these different colors that you see here would each represent sections of the genome that are identical by descent to one another. And what we want to do is to have sequenced enough animals at the top of the pedigree that we would know the sequence of a lot of these colors. It's going to take a while before we'll know the sequence of all of these colors, but the idea is that if one of these colors originated from an animal that was sequenced at the top of the pedigree, then by tracking the inheritance through the pedigree, where we have one of these colors and it's not in one of these kind of fuzzy areas where we have some ambiguity as to which color it originated from, then we should be able to work out the sequence for an animal down here, and we would hope to maybe be able to do that for less than $10. And so we think that's a lot of additional information that the industry needs to be taking advantage of. So the applications for sparse genome scan genotyping are pretty much the same ones that I listed for the seedstock industry above: parentage, verification and determination, genetic defect testing. With the sparse genome scan, there really should be no need to do separate testing for genetic defects. We ought to be able to infer it from the sparse genome scan in most cases, not all cases, but in most cases. Prediction of quantitative traits should be much more powerful through this approach, integrating with the 50K chip and the sequencing, and then testing for single gene traits. So a lot of people contributed to this project. Amanda Lindholm Perry, Linda Flothman, and John Pollack are here at MARC, and John Curry, Paul Deere, Maria Shen, Jessica Nguyen, Slava Fovanov, Nadim Bilsera, Jingtao Liu are at Eureka. Didier Perez is the primary contact person at Eureka. With that, I'll close.
Are there any quick questions?